Arnab, please make me host. I can't record. Arnab, make me host.
Don't worry, it's being recorded on YouTube and Facebook.
अरे हाई क्वालिटी इस पे रिकॉर्ड होगा हार्ड डिस्क पे.
It's 11 o'clock. Good morning on the first Sunday of the month, and welcome yet again to an online Chai and Why. Before we get to today's session, I have a few announcements and some good news actually. From next month or next year, as you'd like, we will begin January 2021 with our sessions back at Prithvi Theatre. But don't worry, we know that you love the online thing. We have so many more listeners, so we will be both at Prithvi and online. But before that, let me give you an update on the sessions coming up. We have another session this month on Sunday, December 20th, which is a day before National Mathematics Day. And that session will have a mathematics theme. And we've requested Ramprasad Saptarshi from the School of Technology and Computer Science, who works on complexity theory, has done a lot of work on COVID modeling, etc. So he'll be speaking to us 2 weeks from now. And remember that from January, we will be hopefully back at our venues live as well as online. We don't know exactly what the topic will be, but I assure you we will start year 13 with a bang. But apart from Chai and Why, there's lots of stuff happening. In fact, every evening, if you go out and watch the sky just after sunset, about half an hour after sunset, the western sky, you cannot miss the two bright objects there, Jupiter and Saturn. And if you notice, every day they're actually coming closer and closer together, and they will almost be on top of each other. The separation will be just 0.1 degree on December 21st. It's called the Great Conjunction. This is happening for the first time this close since I think 1680 something, and it won't happen for many, many, many more years. So don't miss this opportunity. Take a look at this. All you need is just your naked eye. And on the 21st, it's going to be remarkable. They will almost, you will, at least a middle-aged person like me is going to have it very difficult to distinguish Jupiter and Saturn separately. Remember, the moon is about half a degree, and this is 0.1 degrees. And if you have a telescope, you'll actually see a beautiful sight of Jupiter and Saturn together with their rings and moons respectively. We'll try and set up telescopes at different locations in Mumbai with appropriate distancing, etc. But you don't even need a telescope, just go and enjoy this. But you can do more than just watch it. You can make measurements. And all of science starts with measurements. And the Astronomy Society of India, their public outreach committee, they've actually set up something called the Samanta Chandrasekhar Challenge. Samanta Chandrasekhar was one of the great naked eye astronomers from Odisha. And he made very, very simple measurements with a very simple device. In English, you call it the cross staff. All you need is basically a ruler and another smaller ruler and some pins and hair clips and things like this. You can make a device which allows you to measure the separation in angles between two objects. And then it's a matter of doing some very simple maths, similar triangles, stuff that we learn in school. And you can actually make measurements of how the separation between Jupiter and Saturn is coming closer and closer. And then again, it will move apart. And do check their website. You can submit your entries and even win prizes. Remember, we will announce all this and our events also on our social media channels. We are on Facebook, we are on Twitter at Chai and Why. Please do follow us. Okay. So with that, it's time to take a look at today's talk and introduce our speaker. So Ankona has been an integral part of the TIFR outreach team. She's given several presentations, of course, at Chai and Why as well, at our Frontiers of Science programs, etc. She calls herself a scientist who's trained as a chemist, but works at somewhere between chemistry and biology, looking at how to make stuff come alive and watch things inside cells. That's what she's going to talk about, of capturing life in action. And without further ado, I'm going to hand it over to her. I'd also like to welcome Shayoni, whose results, actually many of her results you'll see probably today, has just finished her PhD from TIFR with Ankona, and Smita Rupa, from the same lab, and they've volunteered to look at the questions and moderate the discussion on YouTube and Facebook. So remember that since we are not physically able to see you, the only way we can communicate with you is if you give us feedback via the chat. So please enter your questions, and Ankona plans to make it interactive. So if she asks you something, please quickly type out your answer on the chat in YouTube or in the comment section on Facebook, and Smita, Rupa, and Shayoni will take a look at it and tell Ankona about it, and we'll take it from there. And feel free to ask as many questions as you want. The only thing we can't offer you is a cup of chai, but hopefully from next year we'll solve that also. We'll be back with chai and why. Right now there's no chai, well, unless you can make your own. But let's hear about capturing life in action. Over to you, Ankona.
Okay, hello and good morning, everyone. So, as Arnab has introduced me, I am Ankona, and I'm here to tell you about these special molecules that we make in my lab, which we affectionately call as sensors, and how these molecules can capture life in action. So, Arnab said that we don't have chai, but I brought my chai in a flask. So, hopefully, you also are at home and already have a cup of chai or coffee in front of you. So what do I mean by capturing life in action? Well, we are in Mumbai. So many people might think that she's going to capture Bollywood actors and actresses in action and make a movie. Well, I'm going to do exactly that, but not with the actors and actresses, but with the molecules that are there in a living system, or all the molecules that are there in our body that actually make us a living thing. So here on the right side, you're already seeing a video of capturing life in action. So what we have here is the brain of a larvae, of a fish larvae, and you're seeing the neurons that are lighting up in green inside the brain. And what exactly is happening here is inside the brain cells, calcium ions are being released, and these calcium ions are sending the signals. And we have these smart sensors, or the special molecules that I'm going to talk to you about today, that are inside the brain. And you can see how the calcium is being released from the brain, and perhaps if we knew what the fish was thinking at this time point, we could relate this signal to what it was thinking or what it was doing. And this is exactly what is happening in my brain and your brain and everyone's brain as we are going about with our lives. So when I mean capture life in action, what I want to do is I want to track the molecules of life and find out what they are doing within our body under normal conditions, what happens when we have disease conditions, and so on and so forth. So usually we have a live audience in Chai and Why, and it's really a lot of fun to see the children in the audience. I don't know if we have some today, but I thought we just start from the very basics and tell you about the molecules of life. Well, we are all familiar with what is there inside us. So we have a lot of water, we have metal ions like calcium, we have, for example, phosphate in the bone, and we have, let me just start my laser pointer, and we have a lot of sugar as well. And so when we think about these molecules, of course, these molecules are what make us, living things. And we are going to discuss a little bit about how these molecules are functioning in order to make us alive. So most of the molecules that are there in our body have a lot of oxygen, hydrogen, carbon, nitrogen, and phosphorus, as well as metal ions, as I already mentioned, and a little bit of chemistry, very basic chemistry. So these days we get introduced to molecules and atoms almost in the third to fourth grade. So most of you are familiar with the formula of water, H2O. Sugar, we have 6 carbons, 12 hydrogens, and 6 oxygens. So it is C6H12O6. And you'll see some of these formulae in my talks, because inherently, although I'm trying to understand biological systems, I'm a chemist. Well, when we think about life, we are always thinking organic, and we are thinking carbon, hydrogen, oxygen, nitrogen. But if you look at the periodic table, actually we have a lot of different elements in our body. And for example, manganese, iron, nickel, copper, zinc, these are essential nutrients that we eat every day from the different things that we have. And these are very critical for the functioning of different organs and cells in our body. So these form the building blocks then of life, just like we play with, children play with blocks. So these are the building blocks that make the molecules of life, and that's how life functions. So again, basic chemistry. How do atoms form molecules? Well, they form molecules by either donating electrons or sharing electrons, and we have different representations. So for example, when two electrons are shared between the carbon and nitrogen, we have a single bond, and you're going to see these structures. So pay attention here. I'm not able to see who is paying attention and who is already turned themselves off, but just pay attention here. This is a single bond, and we represent it as a line. And then we have oxygen attached to, bonded to hydrogen. This is an OH bond. When we have two OH bonds, we have H2O, which is water. And so, because we have lots of complicated molecules that are rich in carbon, we can't always show the carbon in all the structures. So we just, join two. This is the carbon that is going to be here. This is just a cartoon representation of sugar, C6H12O6, and you can see that it has a carbon-oxygen bond. So the carbon is a dot over here. This is another carbon bond, and then you have a carbon-oxygen bond. This is a hydroxy group, and so this is the basic chemical structure of sugar. And you're going to see some structures like this in my talk. So hopefully you have, so far you remember your basic chemistry so that you can follow through. So now let us come to, so we have of course water. 70% to 60% of our body has water in it, but we also have other important molecules that are very critical for our functioning, and these are proteins. And where do we get proteins from? The answer is already here. So this is not going to be my Kaun Banega scientist question that is coming in a bit. I'm going to ask that question. But so proteins, we get it from lentils and pulses. And what are proteins made of? Proteins are made of these molecules known as amino acids. Now you can see a different formula, right? Amino acid C11, like this is one formula of one amino acid, C11H12. N2O2. Now you might be thinking, this is all becoming too like chemistry, but I'm going to try and relate this to what you see daily. So these amino acids are joined to each other. There are 21 essential amino acids that we have. They are bonded to each other by sharing electrons, so forming covalent bonds, and they form these molecules which are like beads, and you can see that when you have these when amino acids are joined together, they form these three-dimensional structures which are known as proteins. And so these proteins, these are not this huge as I'm showing in. These are a few nanometer size, and these proteins are very critical for essential functions. So they catalyze reactions. So for example, I'm sure you are familiar with photosynthesis, right? And photosynthesis is a process where in plants, and here also proteins are responsible for electron transfer, for catalyzing the reaction, and so these are very important workhorses of life. Now, so when you of course, when you think of proteins, you're thinking about the pulses and lentils, etc. But amino acids, when you have read the names, there are this, like for example, arginine resist in this hair care product. Arginine is actually an essential amino acid. I don't know how it is helping in making hair better, but you will see that you are actually familiar with many of the names of these amino acids like valine, alanine, and these are parts of different kinds of products that we use regularly. But of course, these essential amino acids are making the proteins in our body, or are parts of proteins in our body, and that is how and the proteins are helping us function. So we have all these molecules. We have millions to trillions of molecules in our body. We have proteins, we have sugar. Now you should be able to identify sugar. We have metals. So if I just take them in a flask, like if I just take them in a bottle, I just mix some protein with sugar, with calcium, and take, let's say, all the molecules that make life. Am I going to get a living system? Although we have the picture of of a person here, that doesn't mean that this is going to be a living system. Why? And that is because molecular interactions are important. If the molecules interact properly, then life functions properly. But then now I'm going to ask the Kaun Banega scientist question. So, I can take these molecules of life, proteins, ions, DNA, whatever, in a beaker or in a conical flask, and they will also interact with each other. Sodium is going to interact with chloride. The protein likes to bind to calcium, so it is going to bind to calcium. But then why is this picture of taking all the molecules inside a flask? I don't have a conical flask in front of me, so I'm just showing a bottle, equivalent to a living system. All non-living things are also made of molecules, and we are also made of molecules and atoms. So quickly type in your chat box, because this is the Kaun Banega scientist question of today, and I'm going to look at the answers. Mitarupa and Shani are going to get me to the answers, and tell me why, if I just take the molecules of life inside a flask and they are also able to interact with each other, why is this not equivalent to life? So I'll take a minute and wait for some answers. Smitarupa, is somebody typing anything? Shiny?
Not till now.
Not till now.
Not yet.
Not yet. Okay. Okay, we'll wait for a minute, for a few seconds, and we will see if somebody starts typing something and telling me why, if I take the molecules of life in a flask, it's not a living thing. Should be obvious, right? Or not? Okay, so
Okay, there is one answer.
mhm.
Yeah, there are two actually. One is Ruhan Patil said the molecules and the atoms need to be moderated continuously to make a living thing working.
Right, very nice.
And there is another answer from Raj that there can be no absorption or dissipation of the molecules.
Okay, entry or exit of the molecules. Okay, we are getting all nice answers.
Yeah, there are a more number of answers coming.
Okay,
Ram Sheshadri,
yeah.
life requires replication. Another came from Darshan Gupte. The arrangements and interactions of the molecules are important.
Very, very nice. So all of them are correct answers. All of you have become scientists now. And so you're right, absolutely. So first of all, we need to take inside the correct molecules have to enter. They have to reach the right place where they are required. And most importantly, as you have correctly understood, that their distribution change, or, with time, whatever is going on, so absorption, dissipation, like removal from the body, and also their location and fluctuation. So basically what you saw in the first video was the concentration of calcium was changing in the neurons. If this concentration change doesn't occur properly, then the brain is not going to function. So change in concentration with time is very, very critical for life to function. And so when these locations and distributions go wrong, right, when molecules are not able to reach the correct location, when their concentration changes are not happening with time, or properly with time, then we have diseases. Or in some cases, for example, the concentration of a particular molecule might increase. So the question is, if I want to understand what is, which a question that has been fundamental to, I would say, both womankind and mankind since, I would say the time when human beings were on earth, is how does life work? And, how do we understand, what went wrong during diseases? So when somebody is sick, of course, what you try to do is you want to look inside in many cases, and you want to see what has gone wrong. In some cases, you will go to the doctor and they will draw some blood, and they are going to try and do some tests to see if your calcium levels are high or low. So now if you're a chemist, you're going to start thinking in terms of chemistry. So you're looking at calcium levels, you're looking at glucose levels. But in some cases, the doctor might want to look within and would say, okay, let's say the body is like you're looking at the globe, and you try to zoom in using Google Earth, and you want to see where is TIFR, right? You want to see where certain problems are happening, and you want to zoom into this particular area. You can see the amoeba garden here, and you can see all the structures, the buildings and the area, right, in a static image. And this is similar to X-rays MRIs, PET scans, and these kind of scans that doctor often recommends when there are certain internal problems in trying to understand what is wrong within the structure, in the internal structure of the biological system. So this is all good, but then, and this is like just getting a still image of TIFR, which I just zoomed in Google Images. But then how do I know what is a specific type of molecule doing, and what goes wrong when the molecule is not functioning well? So what I need to do, for example, is Chai and Y regulars know Kulkarni ji, and he had his office in D406. TIFR, like Mumbai, has very small office space usually, and so Kulkarni ji in 2010 till very recently had an office in D406, and now in 2020 Kulkarni ji is happy because he has a new bigger office for outreach in DM106. So the same person, depending on the location where they are and the circumstances around them, is going to change with time, and depending on the location, their mood is going to change. Similarly, for the molecules as well, the location, distribution, and changes with time are very, very critical to visualize, so that we can see whether the molecule is functioning in a happy state or is doing something that is really bad. And to make this more relevant to a slightly advanced audience, we really would like to understand what molecular distribution changes, let us say, leading to cancer, what molecular distribution changes occur when a virus invades a human cell. If we could understand and see these molecular distribution changes, then we would be able to make medicines, design vaccines, we would be able to find out novel ways of treating disorders, right? So this is why this is so important to visualize molecules and what they are doing in living systems. So now, when I'm talking about visualizing molecules, we first need to, so visualizing means if I were seeing you in front of me, whatever I would be seeing is how I would like to see the molecules. If I'm not there, then I would ask somebody to record a video of what was going on and send it. So if somebody doesn't attend the talk today and wants to come later on, then they can see the video, right? But the problem here is, like, I can do that with a normal like camera that is there in my phone, but about the molecules, they are really not as big as us. We of course realize that. So how big are the molecules? So we first need to know the size of the object that we want to see, and molecules like proteins, for example, a few nanometers. So just to give you an idea of how big a nanometer is, if we just look at the width of this paper that I'm also showing you in the screen, this is about 100,000 nanometers. So you can imagine how small the molecules are, right? So in order to visualize the molecules, then I will need a special camera with very, very high zoom. And here I'm just showing you in a microscope, which has a micrometer, 10 to the power minus 6 meters resolution, which is 1000 nanometers. Using that, we can zoom into the basic unit of life, that is a living cell. Right? And you can see here a transmission image of a cell, where you can see the border of the cell, you can see a bright spot in the center, which is the nucleus, and you can see certain dots, which are possibly organelles or some things inside the cell. So at least with this kind of resolution, we can hope to see several clusters of thousands of molecules in a particular region, and we could see the distribution changes of thousands of molecules. Now with the modern microscopes, we can actually zoom in further, and we can even go to the single molecule level. So this is all good. So we will be able to take the picture, we will be able to zoom in into living systems, and we will be able to see the cell, and we should be able to see, or we will be able to take a picture of the molecular distribution within the living system. So that is all fine, but we were talking about the fact that what differentiates me from the bottle of water, which has all the molecules of life, is that the molecular distributions are not static. So the question is, how do we, look at the distribution changes with time? So for example, let's say we missed this World Cup cricket match, and we had just three photographs, still images. It is very difficult to say who won the match, what happened during the match, how many sixes were there, how many run-outs were there. We cannot really analyze the match and find out who played well, etc. So what we would have to do is we would have to take many images so that we can capture the entire duration of the match, which is going to be essentially making a video. So this is what we need to do with the molecules. The molecules, when they are functioning in the biological system, the processes are happening in milliseconds to seconds to minutes time scale. So we need the ability to zoom in and make movies. And the current modern microscopes allow us to do that. We are able to zoom in and we are able to make movies as well. So I can just see with time what is happening to these cells, whether they are moving or something is moving inside the cells or not. So that is good enough. But then the question is, if I show you this picture, this is all transparent, right? I'm not able to see, how do I know, for example, what molecules are here and, what molecules are in this part of the cell, what is there in the membrane. I really don't have any idea of there's no way of identifying a molecule. So it's the same as, so how do we distinguish? There's of course sugars and proteins and everything here, but do you see something calling out and telling you, I am sugar, I am here, now I am protein, I am there? You don't see any of that, right? So how are we going to distinguish? Well, just like if I put players like of India and Bangladesh, or of two football teams in a field, and if they are all wearing the same jersey and I don't know the players, I don't know who Ronaldo is or something, then I would not be able to distinguish the two teams. So all I need to do is now I need to bring in some jerseys and some other helper molecules that are going to help me distinguish and see sugars, for example, or calcium or protein, right? And are there any questions in the chat so far, Smita, Rupa, Shiny, or we are doing good?
No.
Okay, so far so good. Okay, so people are understanding. Okay, so we want to distinguish these molecules, and the question is, how do we distinguish these molecules from each other? Like, this image, if I could make it like this, for example, where each molecule is showing me an emission in a different color, then I would be able to, say, okay, there's high concentration of lipids here, there's high concentrations of sugar there, or something of that sort. And how are we going to do that? Is there a simple way of doing this, of putting jerseys, colored jerseys on molecules so that I can see what they are doing, what kind of game they are playing inside the biological system? So that is the question. And what we do is we make use of this concept of fluorescence. And many of you are familiar with this concept. Fluorescent molecules are these special molecules which, when excited with light, on which when we shine light, they go to an excited state. And just like, if you give energy, if children eat a lot of sugar, they have very high energy, and then they are going to run around and try to dissipate this energy. Now, these special molecules, when they dissipate their energy, that is when they come down to their normal ground state, they emit energy in the form of light and visible light. So when we shine light on these molecules, when we excite these molecules by using light of a certain wavelength, they emit light of a longer wavelength in the visible region. And we are all familiar with different kinds of applications of fluorescence. I'm going to show you some applications in lab, but of course you can have these nice fluorescent bright shoes when you're dancing, and all sorts of other applications of fluorescent molecules.
Ankona,
yes.
There's a question from Ram Sheshadri in YouTube. How can we be sure that the markers aren't changing the structures we want to study?
That's a very good question. So that is something that you have to, in your design, you have to make sure that the markers do not affect the biological function of the molecules that you want to see. And the other thing is that the concentration of the fluorescent sensors that we will be sending has to be much lower than the actual concentration of the molecule, so that it does not affect the normal function. So it's just like a detector, like very small concentrations. It is sensitive and it detects any change, but the number of molecules of the sensor is maybe 1,000 times or 100,000 times less than the actual concentration of the molecule. Does that answer? I hope that answers the question. Are there any other questions?
No, thank you.
Okay, so we will move ahead. Okay, So with that, of course, a molecular sensor is going to be a molecule that is going to recognize one particular molecule, right? And when we shine light on it, it is going to start giving a fluorescence. So basically what is going to happen is if you have this molecular sensor in a biological system and it sees the molecule of interest, it is going to start glowing and you are going to see an emission that tells you, okay, this is where the red molecule is located in the biological system. You can also have, so we'll come back to this, but then let me give you one very simple example of sensing proton concentration or pH, so in biological systems and how this actually works. Okay, so if I just take so what I'm going to do is I'm going to show you a video of a fluorescent dye in just a small vial, and we're going to add some acid and we're going to see how the fluorescence changes. So this is a sensor which is known as fluorescence. I will just need to escape and play this video. And fluorescence is fluorescent. So here we are shining a UV lamp on this on fluorescence. Fluorescence actually gets excited close to the blue region of light, and I'm adding, so my student is adding acid to this, and you can see that the fluorescence goes away immediately, right? So this is a sensor for pH because we are adding acid. It's H plus is interacting with the molecule, and we are seeing that the fluorescence completely goes away. Now, if I remove the acid, which I can do by adding a base, then we are going to see what happens next, and you will be seeing that the fluorescence comes back on. So this is a very simple sensor molecule that is able to detect pH changes, right? And this is and these molecules are very important because pH changes occur in cancer cells, and these kind of molecules can be used to detect tumor cells inside the biological system. So you can see that as my student keeps on adding the base, we see that the fluorescence is coming back on. Okay, so this is a very simple sensor for detecting just proton concentration changes. This is a molecule that basically interacts with protons and binds to protons, and it changes its structure in a way that it becomes fluorescent. So we will go forward from here. So now, we have detected, or I've shown you a very simple way of detecting protons using a fluorescent sensor. But what about the other elements and ions and molecules of life? So we're going to go from very simple systems to slightly more complicated systems throughout the talk. So I started with pH sensing, and you saw that with fluorescence. Fluorescence is an organic molecule that interacts with proton. It changes its structure. The structure changes in such a way that the molecule becomes fluorescent. Okay, so now we move ahead to metal ions. These are, the next slightly going a little higher level from that. So metal ions are essential nutrients. We have to take them. We don't synthesize in our body, but we take in sodium, potassium, calcium, manganese, iron, zinc, and copper daily in our diet. And what are these useful for? Again, the next question for the audience is, can you give me an example of where iron is present in our body? This is a very simple question. Quickly. Any takers so far? That is going to give me a minute to drink some water. Smitarupa, Shiny?
Not yet.
Not yet. Any example of where we have iron inside our body? Okay, since we have less time, you can think about it. In the meantime, I'll give you the answer. We have iron.
Yeah, there are a couple of answers.
Okay, wait.
So Ram Sheshadri says blood. Vishal says hemoglobin.
Yes.
Blood, hemoglobin, blood, heme, blood. These are the answers.
Exactly. That is what I was going to say. Iron is there inside blood in hemoglobin where it is bound, and I'm showing you a little more complicated molecules. This is a porphyrin ring that binds to iron inside the protein which is known as hemoglobin, and that gives the red color to blood. Very interestingly, these lobsters, for example, their blood is not red in color. The molecule that is there in their blood, hemocyanin, actually has copper in it, and oxygenated blood in the case of these lobsters has a color blue. So essentially, depending on the metal that you have, for example, in the oxygen carrier protein, then you have a different color for the blood, right? So these are obviously essential. Metal ions are very essential for us, and they not only are parts of proteins, but they are also participating in signaling processes. They are important antioxidants. For example, manganese is an antioxidant. Let me just go to my laser pointer. It's an antioxidant present in the brain. The other metal ions function as catalysts for reactions, and so these are very, very critical. And since we are not generating metal ions in our body, we have to eat them. They have to move from the stomach. They have to go to the correct location where they need to function. And of course, somebody might eat more of milk, drink more of milk, and they would have extra calcium. But that doesn't mean that they are going to get excess of calcium and suffer from a disease. Of course, there are diseases where you have excess of calcium. Why is that? In those cases, the excess is not thrown out properly. So in all of our systems, in every cell, there is a mechanism of taking up metal ions, there's a mechanism of removing extra metal ions, just keeping the exact amount that we need. And if any of these mechanisms don't function properly, it can lead to severe disorders ranging from Alzheimer's. Like it's related, I mean, these kind of disorders are related to Alzheimer's, Parkinson's, and some direct metal-related diseases like Wilson's disease, and so on and so forth. So, if I want to make a sensor for a particular metal ion, of course, we will have to think about the fact that the body has all of these metal ions. All of them, as you can see, are cations. Many of them have 2+ charge, that is +2 charge on them. How do I distinguish one metal ion from another and detect it inside the biological system to understand its function and how its distribution has changed with time, right? So the way we try to do this is by very basic chemistry, where we consider, of course, we know that the metal ions are electron deficient, and they are very happy when they get electron-rich donor atoms around them. So we can design molecules which are going to have, for example, nitrogen or oxygen atoms which can donate electrons to the metal ions, and that way the metal ions are happy. But different metal ions prefer a different geometry of the donor atoms that actually stabilize them. So for example, zinc likes a tetrahedral arrangement of these donor atoms. Iron likes an octahedral arrangement. Copper likes a square planar. Also, it can like an octahedral with slightly longer bonds in the axes. Manganese likes like a planar arrangement with 5 donors in a plane. So as you can see already, based on this very simple concept that these metal ions prefer different geometries of electrons, electron donors around them, and that depends on the number of electrons on the metal ion itself. We are not going to go to the details of that, but if you have questions, we can later on discuss that. So if we can just design a molecule such that that molecule has 5 nitrogen atoms in a plane, attach it to your fluorescent dye, and how do we do that? Well, of course, we design it, and then we have to make it, right? So just like my son 5 years ago used to play with building blocks, we also play with molecular building blocks in the lab. We start with simple molecules that can help us synthesize, design and synthesize a molecule that would have 5 nitrogens in a plane attached to a fluorescent dye. This was done by my first PhD student Shubha and Anindita, and taken forward by Aranya, Shani, Kaustav, Smitarupa and Kunika, the new ones who are taking this ahead. So this must not be a very simple job, the simple way that I'm showing it. It's a lot of hard work of the student that goes into designing and making these molecules. But once we make this molecule, if the design is correct and if we have predicted the geometry that manganese, for example, likes properly, then manganese is going to come and bind to this, and you're going to see a fluorescence increase, right? So this is what we were able to develop in the lab. And what we found here is that, when we have, for example, a solution which does not have manganese, and you shine UV light on it or visible light on it, blue light on it, you can see that there is no emission. But in the presence of manganese, we have a very bright fluorescence color. So this means that just like I was showing you the video with the pH sensing, you can see over here that the sensor actually changes color in the presence of manganese selectively. So now let us go to the cell images that I was showing you earlier. So you can see these are the cells, and we have added the sensor. The sensor has entered the cells, and this is like a few microns, the scale bar. So you can see that this is the outline of one cell, and the sensor seems to be fluorescent only in one particular region of the cell. So is this where, the address of manganese is inside these cells, and is what information is it giving us about the location of this metal ion? So you can see in every cell in a particular region, most of them, we have these bright spots, and this is the center, is the nucleus of the cell, right? So what is this address? So in order to explain that, we are going to go to another project of mine during the lockdown with my son. So 6th and 7th grade, I think all of us, I also did this, where we made this model of animal cells and plant cells, and we used this model to explain where exactly the manganese was located. So we did a series of experiments, right? And we found that the manganese was actually located in this intracellular organelle, which is known as the Golgi body or the Golgi apparatus, okay? And we also found that excess manganese, which happens under many disease conditions, actually seems to be possibly leaving the biological system from the Golgi body. So if I keep on adding manganese to the cells, it accumulates more and more in the Golgi apparatus and then leaves. We also found, with some experiments in collaboration with a doctor in UK, that if children have mutations in manganese transport, then they can accumulate manganese in the cytoplasm, and in their case manganese possibly does not leave from the Golgi body, and hence there are the transporters are affected over here, and so you have manganese accumulation inside the cytoplasm, and that leads to disorders similar to Parkinson's disease. So with this, of course, we would like to understand how the metal location changes during diseases, and with the help of these sensors, we can answer questions related to what goes wrong when the metal ion is not located in the correct place. And most importantly, we not only, human beings need metals for survival, bacteria and viruses also need metals for survival. So first things that they do when they invade us is they try to, take up our metal ion pool. And manganese, for example, is very important for immunity against viral and bacterial infection. So it's also critical to understand how the manganese location changes, or the location of this metal ion changes when we have these infections, and how we can stop the bacteria and virus from taking up the essential metal ions from our body, because that could be a way of treating these diseases. So with this, then, so you might say that fine, when we took 5 nitrogens in a plane, and of course this is not bonded together, these are just attached by carbon chains over here. So you don't get confused. I'm not showing a bond between the nitrogens over here. But if I change the geometry, what happens? Now if I put a tetrahedral arrangement of atoms, what am I going to detect? Let us see if you remember from my previous slides. So now we are coming to questions on the slides. So I have shown you different geometries. Can you type and tell me what, if I just make these tetrahedral arrangement of nitrogens, what am I going to detect? Any takers? This is a more difficult one. This is going to be कौन बनेगा scientist at TIFR वाला question. Any takers? Shani, Smitarupa?
Not yet.
Not yet.
Okay, zinc ion, Vishal.
Okay, Vishal,
answers.
you have to come to TIFR to become a scientist. We would like to meet you. Excellent. Okay, so this is what these two people, Tom O'Halloran in Northwestern University had been working on zinc and copper for a really long time, and his postdoc, who is now also herself a professor in University of Texas at Austin, did, and they had this molecule which binds to zinc and is fluorescent. So for the first time, what they were able to do is they were able to see these sparks of zinc during fertilization. So when an egg gets transformed into an embryo, the egg cell actually has these very small clusters of zinc inside the egg cell, and exactly at the point of fertilization, and you're going to see this video. Just give me a minute. See if it goes back to the arrow. Okay, escape. Okay. Yes, and you're going to see this video over here, and we are going to keep on seeing this. So you can see these, initially there are these very small particle like structures which are actually millions of zinc atoms inside within this egg cell, and as soon as at the point of fertilization, all of this zinc is being released from the egg. Now this is actually still an open question, like people know, and this is the first time that these people were able to visualize these zinc particles, these zinc sparks in an embryo that was in an egg that was at the point that it gets converted to the embryo, at that point where actually life begins, right, at that point. So we still don't understand, and what you're seeing here is that this green fluorescent dye is actually marking these clusters of several zinc atoms inside, or ions inside, this egg, and there's another dye which is an extracellular zinc sensor which emits in the red. So as soon as the zinc goes out, you can see that the color is becoming yellow to red, right? And still, there are several hypotheses that these people had given in their paper. And we don't know what is the exact role, but what we have been able to see, these people have been able to see for the first time, is exactly at the time point when this mouse egg is getting converted, it's getting fertilized and is becoming an embryo, we see these zinc sparks releasing, right? And so there are several hypotheses. Zinc is important for the growth of the embryo. It stops the other sperms from fertilizing the embryo further. We really don't know the answers, and this is what is an active area where people are trying to understand. So essentially, so this was this is one of the major I would say successes of these molecular sensors, where we are able to see such processes and such events that are taking place in the biological system. And this is like seeing how the molecules are behaving in life within living systems as it is happening, right? So with this, of course, let us move ahead to a little bit more complicated molecules, slightly more bigger. So we have looked at ions, like we have looked at ions, for example, zinc and manganese. Now let us see a slightly bigger molecules, and can we see them within the biological system. So in order to do that, now we are going to make some Diwali light-like molecules. So we already had these sensors that were initially non-fluorescent, and they become fluorescent when they emit in the green, when they bind to the molecule of interest. Now we have these new kinds of sensors, which are originally, they have a color, they emit in the green, but when they bind to your molecule of interest, they start emitting in the blue. So this is like even more dramatic, and this is of course not just for fun that we are doing Diwali lights inside the biological system. It's important also for quantifying, because if we know the amount of the unbound sensor from the fluorescence intensity and the amount of bound sensor, we can actually find out what is the amount of this red molecule. So I can know the concentrations, exact values, and how these values change with time. So here I'm going to try and bring you to the next level of complexity. We will talk about lipids. So many of you must have heard of lipid profiling, and when you go for a checkup, a regular health checkup, they try to see the level of lipids in the biological system. Now these lipids are playing several critical roles inside the biological system. The most important role, or one of of the major roles of these lipids is that they form the border of the cell. So when I had shown you the model of the cell, you had seen that there's a plasma membrane, right? The plasma membrane is actually made of these interesting molecules which have two kinds of units in them. One is a water-loving, that is a polar unit, and the other is an oil-loving, that is a fatty acid tail. Okay, so how do we show these molecules? We actually show the polar unit in red, and the tails we show as, literally like tails. Okay, so these molecules arrange themselves in bilayers in the cell. So what happens is, you can see that the fatty acid tails, they are all pointing towards the inside because the biological system has mostly water, and the polar groups, both from the outside of the cell and the inside of the cell, they are water loving, so they're pointing towards the water. So this is the phospholipid bilayer, and these lipid bilayers actually have many different kinds of lipid molecules. We have about 10,000 different lipid molecules inside our body. And what are these lipids doing? Well, these lipids are actually not only forming the border of the cell, but they are also helping in signal transmission. Of course, they are making sure that the things that need to be inside remain inside, the things that should not enter do not enter the biological system. And mostly, during bacterial and viral infections, bacteria and viruses have to first compromise our cell membrane and fight with all the things that are there, the regulatory things that we have, to stop foreign materials from entering the biological system. So and of course, the membrane allows the entry of ions like calcium, potassium, and other things that are necessary. So since we have all of these different lipids in the biological system, I'm just showing now half of the cell, and I'm just giving them different colors, right? So all of these polar head groups have different colors, and you can see these have a different distribution in the cell. And what is happening? So we have these thousands and thousands of different lipids, and when a signal, this is not like light, but any signal is received inside on a cell. So for example, in the brain cells, one of the brain cells receives a signal that I want to move my hand, right? What happens? So the first thing that happens is that the signal has to go from outside to inside. How is it going to go from outside to inside? So it has to be transmitted, and so we have these protein molecules which are going to transfer the signal. So I'm showing these proteins as people here. Now these proteins are nanometer in size, and if you have been listening to my talk carefully, you would have noticed that I told you that the cell is few micrometers in size. Now these two tiny proteins which are nanometer in size, and the cell is like 10 micrometers in size. Now these two proteins, when the signal is received at the border, how are they going to know that some signal was received? And the cell is a three-dimensional object, right? It's like it's not like a football, but it's a three-dimensional object, right? And so they have to kind of find each other. And how will they find each other? So this three-dimensional search in a three-dimensional space that is so huge for these proteins gets reduced to a two-dimensional search on the surface of the membrane when the signal is received, because what happens as soon as the signal is received is the distribution of these lipids on the membrane changes. So for example, signal was received, and this is happening at all of our cells, as I am inside all of our cells as I'm speaking in seconds to minutes time scale. Signals are being sent, and the lipid distribution changes. Both these proteins like the green lipid, so they run towards the green lipid, they bind to it, and then they are able to transmit the signal, right? So if this intricate dance of lipids that is going on inside the cell happens perfectly, then we all function properly. If the levels are fine, the distributions are fine, and that is why we would like to measure the levels of lipids inside cells, then we would be able to function properly, right? But if and this intricate dance of lipids is constantly happening inside the biological system, but if this does not work well, then we would fall sick. And that is what it becomes the importance of detecting this lipids and how their distribution changes with time and their localization changes with time. And that is what many scientists around the world are trying to do now. So the question is, how are we going to detect these signaling lipids within living cells and see what they are doing, right? So I already mentioned that the signals are transmitted with the help of proteins that can bind to the lipid head group. So we will learn from biology. So these proteins are able to bind to the lipids, and this is like the green lipid with polar head group and fatty acid tails. Are there any questions so far in the chat or anywhere? or everybody is fine?
There were questions which have been answered so
Oh,
far.
excellent, excellent. I have excellent
Yeah.
students who are answering questions. So this is great. This is the advantage of having an online talk. You can have other people answer your questions. Okay, so you can see. So, how are we going to design a sensor? So we can just learn from the biological system, and we would try to make a molecule that is also able to bind to the lipid and glows in the presence of the lipid of interest. And how are we going to do that? Again, going back to building blocks.
Ankona, there is one question from Ram Seshadri again.
Okay.
What diseases are lipid malfunction associated with? I have never heard any lipid diseases so far.
Okay, this is a very good question. So this is because we are all understanding this, just about, of course, there's a lot of information on this. Lipid malfunctioning, actually, the signaling lipids, when the concentration of a particular lipid increases, that causes cell proliferation. So one of the first cues of cancer is actually the aberrant distribution of a lipid that basically signals cell proliferation. Neurodegeneration, Alzheimer's, Parkinson's, and all of these things, lipid metabolism changes, and and bacteria and viral infections also change the lipid distributions. So either directly the metabolism of lipids is associated with different kinds of sclerosis and other kinds of muscle disorders, but also as a result of these diseases, the lipid distribution changes. And as I mentioned, one of the early, things that happens during cancer is that the signaling molecules in the cell, the lipids start sending aberrant signals that leads to the increased proliferation. Now, these are not the direct causes, but I would say either the change in the lipid metabolism, or that's what we understand now, or in many cases directly changes in lipid metabolism causes diseases. So if you were to ask me what diseases, almost all diseases change the distribution of lipids on the cell membrane and or are related to a changed distribution of lipids on the cell membranes. And that is why visualizing, so far what has been translated to the clinics is just doing the overall level of lipids, right? And so that is why lipid profiling is a basic thing that is done for most of the diseases. But the distribution is also very critical. And if we could catch these subtle distribution changes, we could really understand what goes wrong in many of these diseases. Okay, hopefully that answers your question. So in short, several diseases ranging from cancer, neurodegeneration, infections, bacterial and viral infections are related to changed distribution of lipids. Okay, so I'm going to move ahead from here then.
Yeah, he replied back. Very interesting. Thanks.
Yes, it is. And this is what we are looking at right now. So you can see these building blocks so how are we going to make a sensor for this? So what we did is we looked at the protein, which essentially binds to the lipid, and we said, can we make a very small piece of this protein using the amino acids, like, looking at the region where this lipid binds, like a very small piece of it, and somehow turn it into a sensor by attaching a fluorescent dye. Now, why did we want a very small piece? Because, we wanted these sensors to enter cells, and proteins are usually larger, than peptides, and they don't enter cells by themselves. So, if I add a fluorescent dye, and this red peptide goes and binds to the lipid, it's still going to be fluorescent. So I will not be seeing any difference whether the sensor is somewhere inside the outside the cell or inside the cell or wherever it is. I won't see a difference. So this is what Shamshud Joha and the other students worked on, and how are we going to convert this into a sensor? And here we came up with this idea of using these dyes which are polarity sensitive. So these polarity sensitive dyes have a different emission depending on the dielectric constant. So I already said that most of the outside of the cell and the inside of the cell is like water. And you can see that the fatty acid tails that they are pointing towards inside, and the membrane is more like oil. So the dielectric constant within the membrane and in water is going to be different. And this is a dye that basically changes its emission depending on the environment that it is in, right? So if I could show you here, this is exactly what happens. Okay, let us go and escape from the pointer option. And so you can see that initially we have this dye dissolved in water. It is emitting in the green. We have very low concentration of this dye because it's a very expensive dye. So we didn't want to use extra. Let's see if the video is playing or not. Takes a little, here. And we added an organic solvent which has a lower dielectric constant, and this organic solvent is heavier than water, and you can see immediately the dye actually changes emission to blue. And so here it's shaking, the student is shaking the vial, and you can see that before it was emitting in the green and later on it is emitting in the blue. So with these very simple sensor molecules, then what we did is we made these sensors that can change their emission in the presence of lipids, and then we take these cells to these sensors to living cells, and you can see these are now living cells. We are showing the emission of the sensors as mountain peaks here now, right? Wherever there's high intensity emission, that is the sensors have clustered because there's more concentration of the lipid. You can see the high intensity peaks, and you can see here that inside these cells, so these are now cells, each of these cells are looking like mountains because we have done this very nice colored intensity plot of the microscopy image. But you can see inside the cells we have green emission because inside the cell it has more water. The sensor has entered the cells, it's emitting in the green. In the membrane, the sensor is interacting with the lipid and it is emitting in the blue. So we are able to detect these signaling lipids now.
Ankona, there is a quick question.
Sure.
From Priyatosh Ranjan, but even the property of peptide will change in the different dielectric constant. How do you deal with it?
Yes, actually we want it to change. The peptide changes its structure. So we have studied this in detail. The peptide binds to the lipid head group, changes its structure and dips into the membrane. And actually we use it to our advantage because the change in the conformation of the peptide when it binds to a specific lipid gives us the selectivity towards that lipid. So in this case, the peptide is very flippy floppy when it doesn't interact because we have taken it out from the protein. It's just like a random coil when it is out in the solution. When it interacts with our lipid head group, it attains a structure, or it changes its conformation and then it dips into the membrane. And that is exactly what we use to make these very special sensors for lipids. Okay. So we will move ahead unless there are any more questions.
No more.
Okay, fine. So now let us see if we can see the dynamics of lipids on the cell membrane. So here what we have done is I've zoomed into one cell, and wherever there is mountains which are peaking with green or red or yellow, there's high intensity, which means that the sensor is mainly localized in that region, and we have got these mountains showing maybe thousands to 100,000 of or more lipid molecules in one region of the membrane, right? And so what we are going to do is we are going to now add a molecule that is going to go and start the signaling process, which is going to deplete this particular lipid from the cell membrane. And this is the basis of how neurotransmission happens inside our brain cells. And this lipid, the one that I'm going to show you now, is very critical for cell movement, for regulating ion channels, the entry and exit of ions into the cell, and are related to many disorders. Even most importantly, a variant of this lipid is implicated in cancer and it causes aberrant cell growth. So what we are going to see is, let us see what happens to the peaks that is representing high concentration. So this is one cell, this is the cytoplasm. The color is slightly different over here because I'm doing an intensity map of the bound sensor. So here wherever you are seeing the green mountains, that means that there is high concentrations of the lipid. So for example, here there's high concentration of the lipid, here there's high concentration, here there's low concentration, and there's almost no lipid inside the cell. But in some of the cells, even inside, you can see some peaks. So let us see what happens over here. And you can see immediately that there is a depletion of this lipid from the cell membrane. So this video is it keeps on playing, and you can see initially there are the peaks. As soon as we add the chemicals, you can see that the lipids are getting depleted from the cell membrane. So we are able to visualize this intricate dance of lipids live now with our sensors, and most importantly in our lab we are now able to see how this distribution changes when for example the cell sees a different molecule like serotonin, for example, which is a neurotransmitter, versus a hallucinogen like LSD, or when it sees a drug that is an antidepressant, how these distribution changes and how the cell is able to send signals. And we are also trying to take this forward to see if we can actually detect the changes in lipid levels at early stages of cancer as well. So hopefully what I have shown you till now is that we have different ways, starting from atoms and molecules, how their distribution changes are very important for life. And we can have these turn-on sensors and ratiometric sensors that can go inside living cells and tell us about not only the location of molecules, but how the distribution changes with time. But other than understanding what is going on within living systems under normal conditions and under disease conditions, these molecules also have applications in many other things like drug screening, detecting these are very sensitive molecules, right? In pollution control, in disease diagnosis. And so I'm going to show you one application where this is a sensor which can detect mercury. And this is just a visible color change, absorption based sensor, not a fluorescence based sensor. So you can see that initially the sensor is pink in color. And when as soon as Shiny adds a solution of water containing mercury, you can see that the sensor is changing its color. And we can see the video again. It's pink in color, and it's based on the same idea that you can have a molecule that can selectively bind to mercury, and it can change its absorption instead of fluorescence spectra in this case. And this is not fast forwarded, this is real time, and you can see the change happens almost instantaneously, right? So the question is, can we develop such rapid detection methods for infectious viruses and organisms? I hope we can. We are trying to do some of that research in our lab as well. And so with that, I think I'm going to leave you with this video and try to conclude my talk. It is already 12:05, so that we can get some more time for questions. This is the early days of our laboratory when we didn't have a lab, and how we set it up in TIFR. When all of this pandemic is gone, then hopefully you are going to get the people who became the I think Vishal, right? Kaun Banega scientist at TIFR should be allowed to come and visit TIFR and do some experiments in the lab perhaps. So these are the students working in the lab. That is me. Several years back, I think trying to fix some equipment over there. This is after a lab cleanup, and that is why they're not wearing their gloves and lab coats. And of course, we are not always stuck in the lab. We also have a lot of fun times with the group trips, and you can see that these scientists are also fun-loving people, not like very serious people who are always working in the laboratory. So with this, I'm going to end my talk. I don't know how this 45 minutes or 50 minutes just went. It went so fast. I really enjoyed giving this talk. So we will just end here. Thank you all. I'm going to again get some more chai from my from my flask and answer your questions. So we'll come to both chai and wine. And with that, I'm going to Kulkarni ji, do you want to show the slides first and then take the questions?
Yes, actually, I think we should go through the announcements of the next few Chai and Wai sessions.
Yes.
Yeah. So just to remind you that Chai and Wai will be back in January at Prithvi Theatre on January 3rd. Of course, we will also be online, so not a problem. The next session coming up, though, is an online session on Sunday, December 20th, where we will have our talk themed for National Mathematics Day, which is December 21st, by Ramprasad Saptarshi, who works on complexity theory. Sundays at Jan 3rd, the first Sunday, we'll announce it in a couple of days. Now, don't worry, there's lots of exciting stuff happening in the sky. This month, Jupiter and Saturn are heading to a conjunction on the 21st, and do go outside and check how they are approaching in the evening sky. Just after sunset, you'll be able to see it. The moon will be also there next week to make a beautiful picture. And there are nice fun things you can do. I mean, on the 21st, of course, it will be absolutely together we may not be able to see the separation with our eyes. And the Astronomical Society of India is organizing a lot of public events around this. One easy thing is to make measurements. Yeah. So there is the Samanta Chandrasekhar Challenge honoring Samanta Chandrasekhar, a naked eye astronomer from Odisha. In the 1800s, he had made some beautiful observations of the movement of planets. And you can do this with a very simple thing called a cross staff. Please check the Astronomical Society of India website for details. We will also have it on the CHI-NY page, and we are a part of this thing as well. So go out, try and take a look at the planets, do some measurements, and remember to follow us on social media, both on Facebook and Twitter. Meanwhile, if there are questions for Ankona, it's time for you to put in your questions, and she will try her best to answer them. Thank you for watching CHI-NY, and keep tuning in. But yeah, Ankona, you can take some more questions, I'm sure.
Yes, I'm here. So if there are more questions, we can take some more questions.
Yeah, there's one question from Priyatosh Ranjan. Many of the metal ions possess similar physical and chemical properties. So there are plenty of chances of getting errors, cross reaction.
Yes. Okay, so that is why we have to make sure that the design that we have of the molecules is actually has to take into the consideration the properties of the metal ions. So since Priyatosh seems to be, maybe I can just go back here a little bit. So for example, you can see this molecule here, right? This is the nitrogen macrocycle with the carbons that is actually very selective for manganese. But in this case, because of the interaction of this, the different interaction that this scaffold has with the dye, it is actually sensing mercury instead of manganese. But if I change the dye over here, then I can also make a manganese sensor when manganese comes and sits over here. So there are many design principles that I have not been able to talk about today that go into the designing of these molecules. How, for example, is this binding unit going to be linked with the dye? What is the structure of the linker? What is the position of the donor atoms? What is the size of this ring which is going to fit the metal ion? All of these design concepts, which we do a lot of detailed computational studies in design before designing these molecules. So once we have designed and made these molecules, in order to avoid cross reactions, we first do tests in the test tube where we add different metals and see whether the sensor is actually only responding to your metal ion of interest. Once that is done, then we take it to the living system and hope that it is going to detect the metal. But we don't stop there. We also use other techniques to confirm that the sensor is actually functioning the way that it is supposed to function. So that is why you had these 7 people, 7 PhD students working on this one project, because this happens to be one of the most challenging projects in terms of developing sensors for manganese in inorganic chemistry. Okay, so we'll take other questions.
Thanks from Priyatosh Ranjan, but there are no other. Okay, so there is another question from Shudhangshu Atre. Can molecular sensors be used to understand consciousness, that is self or I-ness, created and disappeared in neural firing?
Yes. Consciousness, I don't know, but yes, we can understand behavior changes. So far that has been done in terms of movement, motion, and trying and understanding which kind of neurons. So mainly the thing is that we have to relate these distribution changes that you were seeing. So if you can go back to my first slide, provided this arrow is going to move. Okay, just stopped a little bit. We'll just wait for a while. So essentially, when we are looking at this calcium signaling firing in the brain, it's not random. It's of course following a certain. It's definitely following a pattern of the neurons getting activated. It's following a certain time sequence in which this is happening. If you could relate this to a behavior or a mood change in the animal, then you would be able to understand, and that is what one of my colleagues in NCBS, Vatsala Tirumalai, does in actually zebrafish larvae, where you are able to correlate these molecular distribution changes with the behavioral change of the animal. Now, whether we have reached the level where we can actually see mood changes, etc., or behavior changes, and link that, because it also we cannot do these in human beings mostly. We do this in animals, right? So whether you can really see these kinds, we can see behavior changes, definitely. We can see escape behavior, we can see predatory behavior and all those things, but whether we can go to the level where we can actually see the mood changes is something that time will only say. Hopefully that answers the question. Are there any other questions? I know there were several questions that have already been answered.
There's a comment, not a question, from Joseph Vinod. Wonderful presentation. Always a fan of Chai and Why, as it helps common public to know what researchers are doing and how it will help the public at large.
Okay, thank you, Joseph, for that very nice comment. We also really enjoy giving these Chai and Whys, and this is like this talk. When I was giving it, just the time just flew. And I really enjoyed myself, although I love having an audience in front of me. Are there any other questions or comments, or should we?
Yeah, there is a question from
There's
Ram
one.
Sheshadri. Yeah, is the future of the sensors to have an app on our phone to give early warning of diseases and such?
There are some people who are trying to link these sensors to apps in phones, for example, not for biological studies, but for example, if you wanted to detect an environmental pollutant, right, or if you were going through an area of high pollution, then if you can have an app in your phone that can somehow give you an information of whether the levels are higher or lower, or you can somehow basically integrate a chip with your mobile phone. Now, in order to, so this app thing, this already, for example, these where you measure the sugar levels, right, using I think an electronic chip where you have to take a little bit of blood, and then so these are basically sensors essentially. Now, whether we will be able to do this now, the problem with us with living systems, of course, which we don't want to do, is we don't want to put many artificial molecules inside us. So mostly the applications of sensors are related to looking at smaller organisms and cellular levels to see distribution changes. But we do use the sensors in MRI as well as PET imaging, where you are trying to detect tumors or aberrations in soft tissue, where you use different kinds of sensors, not fluorescent sensors. But usually the utility of sensors or any molecule that is going to go even drugs, how difficult they are to test. So within a living system, it has to go through a lot of stringent control before we can put any molecule, and we only want to put it if this is a matter of life and death, right, or a matter of health, not just to understand what is going on inside the biological system. For that we use other model systems. Are there other questions?
No.
Okay.
No.
So I think we have had a very nice interactive session. I really enjoyed myself, and I don't know if you want to stop here, Arnab, or Surendraji. They seem to have gone.
Yeah.
Arnab, do we want to end here?
Yes. Can you please stop the live stream and
Okay. So thank
end
you
the meeting?
everyone for this very nice interactive session.